My name is Bob Waters. I am married to Georgiana, who's like in the 6th row back, hiding right now. We have a lovely daughter, a beautiful daughter named Samantha, and I'm 55 years old, and I do not work for UCSF. I am actually I work in IT for a financial services firm here in the Bay Area. But I'm a client of UCSF, and that's kind of what got me here today. So a client of UCSF, what does that really mean for me? So we're going to go back to 2013. I'm going to share a picture of myself so you can kind of get a little flavor of what was going on. So there I am. So in 2013, I was pretty much lost, alone, dazed and confused and terrified. And the reason why was I was diagnosed with bladder cancer. So later in 2013, I was diagnosed with testicular cancer. Then you fast forward to 2015, my wife was diagnosed with thyroid cancer. So we are clients of UCSF. Now I forgot to tell you something else about me. I am a huge Golden State Warrior fan. I mean, I love that team. They're my team, man, for real. I can tell some of you in this audience love the Golden State Warriors as well. 2-0. 2-0 over Portland. Now, as much love as I have for my Golden State Warriors, I got more love for my UCSF team. And the captain of my team is Dr. Peter Carroll. Now, you're going to learn more about Dr. Carroll later today, so remember that name. And he is the captain of my team and clearly orchestrates my care amongst some amazing people, all-stars, I might add. Dr. Amy Lynn, Greta McCare, she's a dietitian that's helped me with, diet, obviously, and lifestyle. Dr. Shinohara, he's worked with us on some stuff. And then Cindy Mock, I really, big shout out to Cindy. She's a nurse for Dr. Carroll. Really, she's like a nurse and a counselor and an angel. She really helped me wrap my head around my journey with care. And just an amazing, amazing person. So where are we today? Well, hopefully you can tell I'm doing a little better, right? And thankfully, both my wife and I are in a very, very good place. Where I was kind of lost and alone in 2013, now I'm kind of part of a team, part of a community that's working with me on my care. We have a plan, we have intent, we're going for outcomes, and that's very reassuring. I'd be lying if I said I still wasn't terrified. I am. And whether I'm dazed or confused or not kind of depends who you talk to. But yeah, so life is a lot better. So the good news for my wife and I is, and we hope we're in this place forever, our situation with the UCSF has changed. Now we're more in this kind of monitoring, prevention, education world. And then that's what UCSF is doing for us. And then we're in a philanthropy kind of role. And I want to tell this quick story. I'm in IT, like I mentioned. I was at a conference. There was a breakout session, much like this. There was a panel group on the stage of all these VC guys, these venture capital folks. Got to the Q&A, somebody raises their hand and says, how do you figure out what to invest in? You could hear a pin drop, man. Everybody wanted to know the answer to that question, right? And the answer was this. We invest in the person that can bring the best talent in the Silicon Valley together to execute against the idea. They didn't say we invest in the best idea, right? It's people. It always comes down to people. When my wife and I donate, it's not like a thank you gift. It's absolutely kind of our investment towards a brighter future. And we absolutely believe UCSF has the people to make that happen. And we are very fortunate to have two of those people here with us today. So I'm going to do my best to say these big words without screwing up. So first, to my far left is Dr. Michael Fischbach. He is an associate professor in the Department of Bioengineering and Therapeutic Sciences. He's a member of the California Institute of Quantitative Biosciences, or QB3. And his laboratory uses a combination of genomics and chemistry to identify and characterize small molecules from microbes with an emphasis on the human microbiome. Whoa. Next, right in front of me is Dr. Cole Roybal. He's an associate professor in the Department of Microbiology and Immunology. He is a member of the Parker Institute for Cancer Immunotherapy, and we heard about that earlier today. He's with the Helen Diller Family Comprehensive Cancer Center, and he is an inaugural Chan Zuckerberg, I think we may have heard of that, BioHub investigator. His body of work has led to several patents and is the foundation of a new company focused on next-generation immune cell therapies for cancer called Cell Design Labs. Thank you very much. Enjoy the session. And Michael, finally, I'm going to turn it over to you.
Okay. Fantastic. Bob, many thanks for that. That was a fantastic introduction. I am going to click over 2 slides to, I have one thing to show you. And I'm here to serve Cole, who will be the next speaker, by trying to add a little bit of context to, and I have very low aspirations. All I am meant to do over the next 2 minutes is to explain to you the entire history of pharmaceuticals from the very beginning up until now, which as you'll see, I'm going to skip a few of the details and just hit the high points. So I'm going to zoom this slider all the way over to the side so that I can make essentially one point. This is an atom of carbon, and if I move backwards from here a little bit, we'll see something that is a chemical. This is an amino acid in your body, but I wanted to show you how big a chemical is. It's made up of a few atoms. The whole history of drugs and the use of drugs to treat humans, most of that history is made up of human chemists making chemicals that solve problems in the body. These are often chemicals that will, in the lock and key sense, get shoved into the lock and broken off and will prevent the lock from going around and causing damage. So this is the entirety of the pharmacopoeia from way back when all the way up until about 1980. Around 1980, something very special happened, and the key events actually happened right here at UCSF. And the thing that happened was that people began doing something very different with the idea of making drugs, but making them not out of chemicals, but out of something else called proteins. And so if I move the slider over a little more, you'll see that proteins, the characteristic protein is actually a lot bigger than a chemical. This is a protein right here. And this is still the chemical, so you can see the characteristic sizes of it. The departure was to say that proteins are ideal for carrying out a lot of the therapeutic tasks in the body. This started out with proteins like insulin and human growth hormone, but quickly became mostly antibodies, proteins that stick to other things. And so now the whole field of biologics, this is Genentech and Amgen are the companies, and drugs like Herceptin, which have been transformative for cancer care, and then some of the newer immune-targeted drugs that people use to treat cancer, some of which you may have heard about already today or will soon. These are all protein drugs. The transformation that's happening now, or the big exciting thing we're here to talk about in this room right now, is that there's going to be a new kind of drug that becomes much more common over the next 20 years that we really haven't seen much of, and that is going to be a cell. You've all heard about a cell. I'm going to zoom way out until I get to, this is a bacterial cell, but really what we want to look at is a human cell like this one. So the use of cells as drugs, I claim to you, is going to absolutely change the practice of medicine. Imagine you had a bolt loose underneath your sink in the kitchen. The use of a chemical or a protein drug to fix that is like throwing a million wrenches into your house and hoping that one of them finds the bolt and tightens it. And a lot of other stuff happens too, much of which is probably not good for you. What you would rather do is to send a plumber in and have him fix the bolt. And not just that, but to say, hey, by the way, I'll stick around for the next 40 years, and if it ever comes loose again, I'll just tighten it and you'll never know about it. That's really what drugs should do, right? They should be very specific, they should be targeted, and they should last for a lifetime. And that's the promise of using cells as drugs. Unlike something that is small like a protein or a chemical. Drugs, or cells rather, can go anywhere. They can do anything. And most importantly, they can make decisions. So the rest of the session is going to be about Cole telling you why it's important to think about how we engineer cells to be drugs, how we get them to make the decisions that we want them to make.
So I first wanted to just tell everyone thank you for your interest in this topic and also for all your support of research. So I'm actually just starting my lab at UCSF, and if it weren't for philanthropy and donor money, I wouldn't be able to pursue kind of these high-risk ideas that we have. And I think these are the things that can really push forward the field of cancer therapy. And I just wanted to thank you for that. So today I'll talk about, for the most part, using immune cells to treat cancer. And I just want to preface this with, if you think about your immune system and getting infected by, let's say, pathogens like viruses or bacteria, usually your immune system can take care of them throughout your lifetime. So you have cells there that can react to most organisms that invade your body. And the nice thing about the immune system is that if you're reinfected with the same sort of pathogen, you have cells on board within your body that then can recognize that pathogen as reinvaded and quickly cure you of that disease. And that's something that's important as far as a feature of your immune system, because now if we can build immune cells that can better detect cancer, they can go ahead and clear the cancer that you already have. But let's say you have relapse. Hopefully you have cells in your body, and this is what Michael was alluding to, for your entire lifespan, and it could get rid of the cancer if it shows up again. Now, when we think about engineering immune cells for cancer, usually I'm thinking about this cell called the T-cell. And the reason why these are good sort of cells to utilize is that they're actually present in our bloodstream. So we can take a blood draw, and then we can isolate this particular immune cell. And they're amenable to genetic modification, so we can retarget these cells so they can recognize cancer and get rid of it. And I'll just show you what these T cells look like. This is through high-resolution microscopy. And this is a T cell that's moving around. So this is another feature that's great about T cells. They can move around the body. This is a T cell interacting with a cancer cell here in blue. And the final aspect of T cells is that once they kind of survey the surface of these cancer cells, they can say, okay, this is a problematic cell or a disease cell, and they can directly kill it. So these cells already have the capability to kill either disease cells like cancer cells or ones that have been infected by pathogens such as viruses or bacteria. So we utilize these cells as a platform to further engineer them, and we want them to better be able to detect cancer, because if our immune system was already good enough, it would have cleared the cancer before it even starts. So how do these therapies work? This is just a cartoon form of a T cell. In this case, we removed it from the cancer patient. And now we modify the T cell with different kind of approaches to genetic modification. And the main thing we want to do is, at least at this point, is give these cells a new, what's called a receptor. And these are proteins on the surface of the cell that will allow this T cell to detect the cancer. And not only does the receptor allow the cell to detect the cancer, but once that detection occurs, it activates the T cell to kill the cancer cell. And then it also leads to this situation where you get multiplication of these T cells and expansion. So you have a bunch of them, and you have an entire army that kills the tumor. So once we've modified the T cells, what you can do, you infuse these cells back into the patient, and then they can move through the body, detect the tumor, and then kill it, okay? And the thing I want to point out is these therapies are working in certain contexts. So there's certain malignancies that these sorts of therapies are curing patients that would normally have no other options for therapies. But as with any therapy, you have kind of common problems and pitfalls, and we need to innovate in this area. So first of all, our immune system is very powerful, and these cells can get in there, and depending on the size of the tumor, there can be a ton of inflammation in your body, and you have to be monitored in the hospital because the overactivation of the cells can lead to toxicity and death in some cases. The other problem is that sometimes these cells detect the tumor and kill it, but then sometimes they also detect certain tissues in the body, and if it's a vital tissue, it can also cause toxicity in those tissues, and that's what off-target toxicity is. And then finally, with solid tumors, compared to ones that are kind of in your blood, these cells sometimes have trouble infiltrating these tumors and really killing the entire tumor mass. So we need to do two things. We need to make these therapies more controlled and safer, but then we also need to enhance the capabilities of these immune cells so they can deal with difficult to treat solid tumors. Okay. So this is one of the first kind of problems that we've addressed over the last couple of years, and that is you have a therapeutic T cell here that has a receptor on its surface that detects this red feature of the tumor. Okay? And if this cell only detects this red feature, it will detect the cancer cell, but then maybe there's a lower level of this protein on other tissues. So what happens is this T cell kills both of these things, and often this can lead to mortality, depending on what the bystander tissue is. So what we've tried to do is now build T cells that can recognize multiple antigens, or multiple features of the tumor. So now instead of having a T cell that recognizes one aspect of the tumor, you build a T cell that has multiple receptors, and only when the T cell sees some combination of features do you get the activation of the T cell. So now you have better discrimination of the tumor versus the off-target tissue. Now this is a little video of what I've actually done. So over the last couple of years, we built some new types of receptors that we can put in T cells. And what you can see is that this T cell, when it goes in the body, has one receptor that detects the green feature of the tumor. And only when this cell sees that feature does it upregulate the production of this secondary receptor that binds this red feature of the tumor. So you need multiple features in order for this tumor to be cleared. And then this T cell leaves alone the different bystander tissues in the body. So we've done this in human T cells. We've done it in mouse models. And now we're moving this work into a company and then hopefully in patients in the next couple of years. So this is a major thing that this sort of therapy does for us. And that is, if you have cells that can recognize multiple features of tumors, a lot of times this kind of single feature approach that people are doing at this point, it would just be too toxic. You would be getting off-target killing of these different tissues. So now what we think is that by using this kind of multi-receptor approach and these sorts of T cells, we can address different tumors that would not normally be addressed by this sort of therapy. And we're moving into things like glioblastoma that are very difficult to treat and don't have great treatment options. Now the last thing I want to talk about, and this is a whole nother kind of area, but it's related, and that is just getting immune cells to be smart pharmaceuticals. And the overall idea here is we want immune cells that we engineer to be able to detect diseased tissue and then produce some of these therapeutics that Michael was talking about only within the diseased tissue or the tumor or whatever it may be. And the reason why this is important is because let's say you go in and get one of these small molecule drugs or one of these protein drugs and they give it to you IV into your bloodstream or orally or something like that. Often there's toxicity associated with that, giving it to the whole body. So if we could actually have cells detect these diseased microenvironments or these diseased environments and only produce the therapeutic at the site of disease, it could relieve toxicity, and it could have a second effect where you could get large amounts of the drug at the site of disease, and it could be a more potent therapy that would be more effective. So the receptors that I've built over the last couple of years, we can put them into cells. This would be a cartoon example of a therapeutic cell. This could be a tumor cell. Once these cells come together, as you'll see in the video, this receptor will drive the production of, in this case, an antibody drug. And this is akin to having things like drones bringing different packages, very select packages. This is an example of a T cell recognizing diseased tissue within a kidney and then only locally within the kidney producing these antibody therapies. And as I've already said, what this allows for is kind of targeted delivery, preventing sort of this systemic toxicity and likely a more potent sort of therapy. This kind of reiterates this idea. We would like to be able to use this as a platform and do this with many different diseases. The cells go in, they detect the site of disease, and then they produce therapeutics. And you can imagine building cells for things like cancer that detect tumors, autoimmunity like type 1 diabetes, where a cell recognizes the autoinflammatory site in the pancreas, or tissue damage or injured arteries. Once they see the site of the disease, they simply produce the therapeutic, and then you have kind of a stronger therapeutic activity. And with that, I think we can go on to questions, and hopefully we can have a kind of discussion around where all of this is going in the next 5 to 10 years or so. Because as Michael said, this is an exciting and new area of therapeutics.
So while we're gathering up the questions, just real quick, can both of you share how you got into this field and what brought you to it? And then also, what brought you to UCSF? What was your journey here and why?
I'll take the second question first. What brought me to UCSF is, I think, when you're on the job market, you have multiple options often, and so you're ending up choosing among places where the research is strong at one place, let's say on the East Coast, and the research is also strong at another place, let's say UCSF. And I think that what differentiates UCSF from other places and what makes it such an amazing and happy institution to work at is that if you come into the room at UCSF with a crazy idea, people will just say, wow, what if we could do that? Let's find a way to make that work. Where the response elsewhere is often people turning up their noses or mumbling and grumbling. So I think that the can-do attitude and the fact that this place celebrates the kind of irreverent, crazy new work that's very likely to lead not to incremental advances, but to wild, new, crazy advances like the ones we've been talking about today. That's what is so special about it. It's also the kind of place where somebody like Cole, who trained as an immunologist, could come and learn something completely different, like how to do cell engineering, because the list of people in the entire world who are great at immunology and also great at cell engineering is very small, not much larger than one. So a place that sort of celebrates doing crazy things like that is really the only kind of place for me.
Great, thanks.
So I would agree with that as well. So I did my postdoctoral training here in a lab that's just upstairs, and I would say that there's this unique aspect of UCSF as far as the collegiality. The immunology department here is second to none in quality, but if you look at the interactions within the department, they're vast. And I think what I found throughout my career is every time I interact with a variety of different people, my work gets better. So I don't want kind of an insular scientific career. So that's one of the reasons I stayed at UCSF. And then, I feel like we're on the cutting edge of this sort of area of cell therapy. And earlier in the talk, the Parker Institute and the Chan Zuckerberg initiatives and all the stuff that's happening in the Helen Diller here, it's just a really unique environment that I couldn't find in Boston or elsewhere. So it's really exciting. And then as far as what got me into this, In my graduate work, I was an immunologist, so I really studied how T cells function, but in a natural sort of scenario, trying to understand how they recognize pathogens, these sorts of things, and how they activate. And one thing that did happen during my graduate school is I had a fellow grad student that was in the lab next door that was very young that died of acute myeloid leukemia during grad school. And I saw that happen, and that was incredibly scary. And then, you know, I thought about cancer research, and coming here, I got into the area of now engineering immune cells. So instead of studying their natural function, how can we change their capabilities to be able to detect and treat cancer? And Wendell Lim's lab here, I feel like provided the perfect setting. And I feel like after seeing some of the instances of cancer in my family and other people around me, I'm excited that I can apply all my kind of, you know, studying the fundamentals of cellular biology, but then also having some applications that could be really important within, you know, years instead of many decades, which is exciting.
That's awesome. So if we had any concern about getting questions, forget it. We're loaded. So we certainly want to give these questions their due, but we're gonna try to move through these and not mess around. So the first question, you are gonna have to decide who to ask because, again, I won't know most of these words. So what are the major bottlenecks for development of cell therapies?
Yeah. So there's many bottlenecks because this is a multifaceted sort of thing that we have to address here. So I was at a Parker Institute meeting the last couple of days, and you know what we're trying to do is not only do you have to figure out, so I had these colors, right? The tumor was designated by a red and a green feature. So we need to use large-scale data analysis on many different types of tumors in order to find out those features of the tumor that we can target. So that sort of thing has to be developed. Big data sort of analysis is something that Silicon Valley and this institution does well. But it has to come together. Then obviously we have to do all the engineering as well of the receptors to be able to detect these different aspects of the tumor. And when I talked about solid tumors, we have to understand what is happening in a solid tumor that is really shutting off the immune response. And that's going to be different for each solid tumor. So that's a huge amount of work as well. And this is why we need to be able to fund these large sort of consortiums of people that can address these different aspects. And then, you know, as far as bottlenecks for actual development of the therapy, since these are new sorts of therapeutic approaches, you have to have the infrastructure to be able to take cells from a patient, manufacture them in such a way that they're of sufficient quality to go back in patients. So that's a whole new thing as well, and there's companies doing it, and there's universities doing it, but that's a huge investment that has to be made.
But let me add on a more hopeful note that I'll just take one of the challenges that Cole referred to at the beginning, which is the challenge of being specific about what tissue is targeted. I'm completely sanguine about where this is going, if only because we already know that cells are capable of doing this. All the time in your body, without you knowing it, cells are going around to one or another tissue, very specifically, only there, and doing exactly the kinds of things that we would like to have them do at our will. They're cleaning up problems by gobbling up stuff that shouldn't be there, by repairing tissues, by calling other cells into action. We know they're already capable of doing this. So it's not a question of will we be able to call them into action the way that we want to. It's just a matter of, it's an engineering challenge. So the question of whether we'll get there is, I've sort of absolute certainty we will. Cells are the right material to be working with. It's a challenge worth taking on because we know that this is a composition of matter that's capable of doing everything that we ultimately will want it to do.
Okay, great. You may have touched on this a little bit, but what would help bring down the cost of therapies and make them more widespread?
I think, So the major thing is there has to also be technology development in the sense that at some point you would like a closed system to be developed in which you can simply take these cells out of a patient. The cells go through some sort of process and they can go right back in. And there's companies developing that. There's technology being built at universities. So you don't want to have this situation where you're constantly having to take cells out of a patient, ship them somewhere to be processed in a very specific place in order for them to be of sufficient quality to go into patients. So we need these sorts of systems to reduce kind of costs of therapy. The other thing is that there may be a situation, or it's already happening, where we can have what are called universal donor cells. So I have cells that we could engineer that could go in any person. Currently, we would have to engineer cells that come directly from the patient, and that would make things cheaper.
Yeah, I would agree with both of those things. The major departure for cell therapy will be that the way that Cole engineers cells right now is to use a neutered form of HIV, a virus called a lentivirus, to insert genes into the cell. And the fact that we use viruses to get genes into cells is itself quite an impressive way of editing the genomes of those cells, but also indicates that maybe one day we will never have to take the cells out of somebody's body. We could just put a very specific engineered virus into the person's body, and it will find the cells it's supposed to go to and put the genes in. The cost of that, the cost of actually manufacturing a virus like that would be similar to what you would pay for a vaccine. So very cheap, much lower than the cost of goods of making an antibody therapy, for example, and obviously much easier to administer. So the challenge of engineering something like that to be safe and specific is large, but you can imagine that this is worth taking on, because if you could have what is essentially an extraordinarily sensitive and specific and effective cell therapy for the cost of a vaccine, this is a no-brainer that this is something we must do.
Great. What are the strategies cancer cells use to defend themselves against the immune system and cell therapies?
Okay. So when I talk about solid tumors, there's a whole host of things that occur in tumors that they shut off immune cells and prevent them from activating. And you can go through a list of probably 5 to 10. They're producing molecules that actively suppress the activity of the cells. Tumor cells can have these kind of receptors also that interact with the immune system, and these receptors, or the things that the receptor bind, can also shut down the activity of the immune cells. If you think about a solid tumor, these are growing rapidly. They're very, what is called hypoxic, meaning there's low oxygen in tumors. And a lot of different cell types don't work well in that sort of environment. So once cells infiltrate into these regions of the tumor with very little oxygen, they're not functioning. And then another thing is that this hypoxia that occurs, or low oxygen, is often due to there aren't many blood vessels getting into these tumors sometimes, and you need these vessels to help get immune cells in. And there's other structures that kind of coat the tumor and other cell types that actively prevent immune cells from infiltrating. So it's kind of a multi-pronged sort of system of suppression. And we have different ideas of how we can build in a bunch of capabilities into immune cells in order to help shut these things off.
All right, next, how do engineered T cells interact with the body's own immune system, which is also producing cells to attack tumors?
Yeah. So I mean, as an immunologist, I actually think you need both things to occur. The engineered cells will be in there within the tumor, as well as your natural T cells. So let's say I take out a biopsy of a solid tumor. Often you have T cells that are within the tumor. And if you do certain assays on those cells, what you find is that those T cells are reactive to the tumor cells and can activate. But what's happened is they're there, but they've been shut off, right? So the idea here would be, can we get engineered cells into the tumor to help jumpstart the immune response? Because you want both things happening. You want the engineered cells to help get rid of the tumor, but then you also want the natural immune response to come in and help clear things.
Yeah, I think that's one of the most exciting things a cell can do. In addition to all the things it can do itself, it can call its friends in, or it can tell its friends to go away. And both things can be very useful. So that, as anybody who is a manager of other people knows, there's only so much you can do yourself, but your team, and if your team consists of managers that manage their own teams, the number of direct or indirect reports you have as a cell is quite a force multiplier. So that's how a very small number of cells could lead to a large biological impact on a tumor, because you can just tell a cell to call its friends.
How you guys holding up?
Great.
Well,
All right. Wow. Will this new cell science replace chemotherapy?
Yes. So let me just say that when they...
Yeah, Cole, what do you think? No, sorry.
When they write the history books, there are books that have been written even recently, The Emperor of All Maladies, these fantastic, amazing stuff that everybody should read if you haven't already.
What is it?
The Emperor of All Maladies, this book about cancer, extraordinarily well written. They will, before long, write a new history of cancer, and of other diseases, I'm sure, too, but we're here to talk about cancer, that in which people will look back and say, it was extraordinary how we used to treat cancer. It was, the brutality of using something like chemotherapy is just crazy. It is not the point of what I'm saying to try to say anything negative about the developments that led to chemotherapies and the number of lives they've saved. That is incredible and amazing, because before that, the situation was much worse. And yet, the idea that we treat disease by using such a blunt tool, I think people will look back and say that that was completely crazy. I can't believe that ever happened. In order to get there, we need to figure out how to use something like a cell. It just makes much more sense as a therapeutic. This is not just for cancer. Other diseases this makes a lot of sense for also. Any disease where taking something that goes throughout your whole body can cause problems elsewhere in addition to solving a problem in one place. But the development of cells as a specific therapeutic makes eminent sense for essentially all diseases of humans.
Yeah, I would agree.
Next up, which cancer fighting approach is showing more promise of success? Starving cancer cells or tumors of nutrients, or mobilizing the immune system to defeat the tumor or cells?
Which one? I mean, I think both things could be important. I mean, one aspect of what we can do is have cells go in there and help to starve the tumor of nutrients, right? So these are things that we can do. So this is one type of approach to, let's say, a solid tumor that is taking up a bunch of nutrients, getting immune cells to now take those nutrients away, and then at the same time help clear it.
Yeah, and I'd say that in professor school, they instruct you to answer every question like that, A or B. The answer is both. And cells are especially convenient for that because, and this is a serious point, we shouldn't have to choose between two things like that. Shouldn't we have a therapy that can do 2 or 3 different things at the same time to help kill a tumor cell? And maybe the tumor cell gains resistance to one of those 3, but not the other 2. That's the way it should look. It should not be that we have to choose one thing, and if it works, great, and if it doesn't, too bad. So the fact that a cell is capable of doing 2 or 3 or 5 or 10 things at the same time is, again, another strong argument in favor of using that as a platform for therapeutics.
And by the way, that's what our immune system does. You have many different immune cell types that have to go throughout the entire length scale of your body in order to get rid of pathogens. And they coordinate behavior, they do all these things. They're doing 20 things at once.
Do you collaborate with researchers from other universities or drug companies?
Yeah, absolutely. So things like the Parker Institute, it's actually, it's UCSF, Stanford, it's Memorial Sloan Kettering, UPenn, also I think Washington and others, so University of Washington and Fred Hutch. So this is bringing in some of the best people that are in this field. We definitely collaborate, and I think that's the only way to make huge impact in these areas. And then also helping to develop the infrastructure and ability to do this sort of work across different institutions, right? So if you're starting to run clinical trials and treat patients, you want to have some standardization across all these places. So we are actually trying to maximize and do that all the time.
Yeah, and then I would add to that that collaboration with industry is also essential, especially with something like this. It's hard to manufacture a cell therapy. It's hard enough to manufacture a chemical or a protein drug. Cells are several orders of magnitude more difficult to manufacture. Just that challenge alone would suggest that it's good to get a company involved, just a group of people who are focused on figuring out how to do that the right way, and even how to take something that has reached sort of the end of what academics can do with it and refine it to the point where it really will work dependably. That's the work of companies. And so although we all know that working with companies can raise the specter of a conflict of interest, here that's something that needs to be managed, not avoided, because if we ever want this kind of work to see patients, it is going to have to go through a company. And so that's something that we embrace as part of the trail that takes us from a basic discovery to something that's actually saved lives.
Is a person's weak immune system what allows cancer to grow? If someone has a strong immune system, would cancer be killed without treatment?
So as you progress with cancer, I think depending on the type of cancer you have, your immune system can be weakened because the cancer is taking over your body. So that's something that does happen. I think no matter how strong your immune system is, certain cancers, they have all these things that we've talked about that to suppress or prevent the immune response. So there probably are a few individuals for certain types of cancer. There would be called like elite responders. So they would have an immune system that could take care of it. They maybe happen to have the right T cells or something like that. But for the most part, even strong immune systems are thwarted by cancer.
So much research is focused on the top 5 cancers. How do we get to focusing on non-top 5 cancers, e.g. esophageal or gastrointestinal? And I'll add my own piece to that puzzle. Is the work that you're doing, is it horizontal in nature or is it vertical against specific cancers? But
I'd say that for the classification of cancer types into lung cancer and breast cancer and ovarian cancer, this is an artifact of the way that the disease has been studied throughout its history. One of the advantages of doing any kind of targeted therapy, not just cells, but of biologics and especially the chemicals that have been developed recently, where we know exactly what they're targeting, is that we tend to think now of cancers less, and you're not a lung cancer person or a breast cancer person. You're more a person who understands how to target one class of signals that, when they go awry, can turn a cancer cell on. That might be something that spans a bunch of different cancer cell types. I think that, again, one of the promises of using cells as therapies is that we can program them to do, I would imagine, almost anything we want them to do. And so something that works on a breast cancer should work if we reprogram its targeting capacity but leave everything else the same, just as well on glioblastoma or bladder cancer or really anything else. And so the generality of being able to retarget immune cells to whatever we want them to target is one of the great advantages of using this as a platform.
I mean, if you look across a lot of the cancers and some of the solid tumors and pull them out, you find infiltrating, let's say, T cells or other immune cell types. we can at this point go in and ask, what are those T cells seeing within the tumor? And we could do that for many different cancers. And then we can, once we know what those T cells are seeing in the tumor that's activating them, we can also do this sort of engineering on top of natural T cells that are able to detect cancer. So we could make a bunch of different products, hopefully, for a wide spectrum of cancers.
You use viruses to program the T cells. How?
Want me to go?
How much time do we have?
It should be only a few sentences. So as Michael said, we use this kind of what's attenuated or neutered HIV virus that, at least that's what he said. And there are different sorts of viruses we can use, but basically what we're doing with the virus is viruses have their own like little genome, right? And they can infect cells. And these viruses infect the T cells that I have in culture, and then they simply add the DNA to the genome of the T cell, right? So if I want to put in the gene for this receptor I want the cell to express, the virus integrates that into the T cell's DNA and genome. And now once that virus is gone because it doesn't replicate, and the T cell now has that gene in it, and it can express the receptor. So that's how that works.
Yeah.
Not 2 sentences, but you know.
And to that I would add there's huge room to improve that technology. There are bigger viruses that could add many more genes. So instead of adding one thing, you could add 20 things to the cell. There are ways to make it so, like we were discussing before, you wouldn't have to take somebody's cells out in order to put the genes in using a virus. And so that is the crux of this technology, and consider it a place where you'll see the most rapid technology development and the fall in price that will accompany that.
So this next question is from the same person who did the previous one, so keep that in mind. I don't know if it's related. When will you use other healing mechanisms of immune systems to hasten regrowth or regeneration of tissues?
So a whole other aspect that I didn't talk about today, because it's more a cancer talk, is you can engineer cells for regenerative medicine, right? So we could take stem cells or other cell types. There are a variety of immune cells that are involved with surveying the tissue and then helping to elicit kind of repair of tissues. So we could engineer those sorts of cell types. They wouldn't be producing the same sorts of therapeutics. They would be producing molecules that can help to repair a wound or whatever it may be, or problems in a certain region of the brain or whatever, if you have neurodegeneration or something.
Yeah, this is stock and trade of cells is repairing stuff. You're getting stuff repaired all over your body right now. The cells are doing this constantly. So the engineering cells to repair the things we want them to repair and doing it with gusto is not something that's going to be too difficult to make happen, but it hasn't happened yet.
Yeah, and Cole, you mentioned the brain. So how close are you to curing brain cancer? I mean, what's in the space?
I mean, something like gliomas or glioblastomas are some of the worst cancers to get, right? There aren't great therapies. I wouldn't say we're close to curing it, but I think the cell therapy ideas and things that we're pushing forward right now could actually have a major impact. One thing about cancer is if you look within a tumor, not all those cells are the same. And we have to figure out how to build cells that can deal with this sort of situation where you have what is called heterogeneity in the tumor. So when not all the cells have the same features. And we're building cells that are able to deal with this sort of thing. And this happens a lot in glioblastoma or gliomas. And we're starting to test that sort of stuff in preclinical models.
Yeah, there's a question that I saw come across my lap about the blood-brain barrier. T cells can cross the blood-brain barrier. So this is that's a barrier of other cells that sometimes keeps chemicals or proteins from entering. Here, cells naturally get into the brain, immune cells, and so that wouldn't be a barrier to curing a glioma or a glioblastoma.
Why not deliver activated T cells directly into the tumor by injection, as an example?
I mean, this is something that can be done. I think most of the patients that are being treated with these therapies are quite late stage and don't have other treatment options. So what that means is that the cancer isn't localized. It has metastasized and it's disseminated throughout the body. So just like in an immune response, maybe you have infections in different parts of your body or the bacteria has moved around. The preferable scenario is that you simply inject cells in the bloodstream and then they can find the sites of disease because they're generally not localized.
Maybe what motivated that question was the concern about can these engineered immune cells get to where they need to go inside of the tumor? And Cole had referred to things that could keep them out. Tumors will sometimes grow kind of a thick coat that will keep cells out. One of the exciting things you could imagine is that cells are also capable of breaking down tissue, fibrotic tissue, and so if the we probably wouldn't have to inject them right into the tumor. We could just give them a clipper and tell them to go to the tumor and start cutting until they get into the juicy part.
Yeah.
What types of cancers have you successfully fully engineered?
You mean cells like targeted?
Sounds like it, yeah.
Yeah. So at this point, the major therapies are more for what are known as B-cell malignancies. So that's a type of immune cell, and these are hematologic, so they're systemic in the bloodstream. And that's where these sorts of therapies are making huge impact right now. So those products are working really well. There's companies like Kite that's down in Southern California that's having great response rates in trials. And then the other stuff is really ongoing. So we built a lot of different, or we've engineered different receptors and different platforms for a variety of other cancers. And over the next 5 to 10 years, we really had to push on it and make this a viable approach for a whole spectrum of things.
All right, what is the T cell kill mechanism?
So what actually happens at that, so when you saw that T cell kind of latching on to a cancer cell, what's happening is it's delivering these little pockets of the cell, they're vesicles inside the cell, to that interface between the T cell and the cancer cell. And it releases these vesicles into the cancer cell, and there's all kind of enzymes within those vesicles that drive the death of the target cell. So that's what's actually happening in that picture.
It's a very robust, hot response.
Yeah.
So how T cells are highly effective. One T cell can go kill many, many cancer cells. It just goes one to the next and delivering, dumping these little bubbles of death. Yeah. That's great if the T cell is doing exactly what you want it to do, but it's also the reason why you have to engineer these to be highly specific, because if the T cell accidentally latches onto a cell you'd rather keep around, that's going to die just as quickly.
Yeah, that video you shared was like stunning. I mean, that was amazing. Any relationship to engineered T cell that injects chemo, therapeutic agent? Does that make? Maybe I didn't read it right now.
No, that's an interesting idea. Naturally, you wouldn't know the T cell wouldn't make a chemotherapy of its own, but there's no reason why we couldn't engineer T cells to produce not just the normal death-inducing things that they make, but other chemicals that might be more specific or might not even kill, but will just alter the function of a cell, tell it to stop causing trouble if it's causing trouble. So I think that. So I'll answer the question by saying it's a nice pivot from and a reference to something Cole mentioned, which is that we don't just have to do death. That's going to be nice for cancer. Most of the applications are finding a tumor and killing it. But if you're a normal human being, you have other diseases yourself or in your family. And so for something like autoimmune disease, it's not killing we're after. It is encouraging other immune cells to stop attacking you, the person, and that's something that an engineered immune cell could do equally well.
All right, I'm going to go 2 more questions, despite what they're telling me. So, any human trials using this new engineered targeted T-cell approach?
So the original forms of the therapies, so when some of the engineered T-cell therapies that first started, actually the receptors were developed here in the late 80s, and then now we're finally seeing these things being used in cells in patients now. So when I talked about the KITE trial, it's really based on this research that came out a long time ago. I think we're at a point where we can do this more rapidly. So that's the major application that's in trials, the one against B-cell cancers.
All right, this is the final question. How do, and I think it's CAR-T therapies compare to bite-based therapies? And how do you say that?
I know what they're doing.
Okay. And are CAR-T more effective in patients?
Yeah. So I have to explain the concept of bites. So, bites are bispecific antibodies. And the way these work really is that it's this protein that binds the T cell, but then the other half of the protein binds the cancer cell. And what happens is if there's a T cell in the tumor, the bite will bind the T cell and the tumor and redirect the T cell so that it now kills the tumor and activates it. Okay? So this is a way of redirecting the specificity of T cells so that they can actually engage the tumor and kill it. And it's effective for certain things. And actually, I've done some work in recent stuff I published where I can actually get engineered T cells to produce bites within tumors. And that works really robustly. That way, you're not giving this systemically in a patient. As far as their relative kind of effectiveness, I think it's going to depend on the type of cancer. That's what I was saying.
This was great. I had a lot of fun.
Yeah. Thanks so much.
Yeah.